So yeah, as I was saying before, thanks for coming. Sorry about that interruption, and I'm glad you made it back. And also, some of you have come to other talks I've done on the human microbiome, so you will find some overlap here, but something's new. And I'll just jump into it, since how we're a little behind already. Probably you've all been seeing a lot of different news items and articles and things in medical news on the microbiome for the last several years. And it's really everywhere. So if you start looking for it, you will see it everywhere. And there's, of course, a lot of people trying to make money off of it, and, microbiome creams and even microbiome toothpaste and, all the probiotics. And some of this I think is really valid. A lot of it probably isn't, or at least not at this point. I don't think we know enough for a lot of these products yet. So hopefully in giving this talk, I can kind of acquaint you with what the microbiome is, how it's helpful to us, and how has our environment changed that's been affecting it, and and how that's led to a lot of the diseases and things that we're seeing higher rates of now than we used to. So first, just what is the human microbiome? And kind of in a nutshell, it's these trillions of organisms that would include bacteria, viruses, and fungi. And I have bacteria bold because that's the most of these microorganisms. If we're talking about bacteria, there are a few viruses and fungi that are considered part of our microbiome that live in and on us. And the bacteria, as it says, predominates your microbiome, where we have at least as many bacterial cells as we do human cells. So we're at least as much bacterial as we are human. There was not that many years ago where they thought it was like 10 times as many bacteria as human cells, but that's been refined as research has gone on and realized that it's closer to a 1-to-1 ratio, but probably still about 10 times more genes from the bacteria involved than our own genome. So anything we do, anything that's affecting us, we have to also think about how it's affecting these organisms that are really very much a part of us. Okay. So where does the microbiome live on humans? And even this has been kind of evolving as the research goes. Certainly we know it lives all over our skin. It lives in our mouth and nose and eyes, any of our, where our mucous membranes are. We used to think the lungs were sterile, but we realized that there's definitely a microbiome there now too. The biggest microbiome is in our colon, but there's also some in the small intestine and also the stomach, also the urogenital tract. So a number of places around the body, but not absolutely everywhere. So one of the things that we've learned is that there's different types of bacteria, so different species and genus of bacteria that have evolved to live in different places on our bodies. And so we would expect to find certain types in certain areas. And then those interact with each other and also to microbes that we contact in the environment, as well as our own cells in really very specific ways. And there's a lot that we've been learning about that, and certainly a lot more to learn. But one of the things we certainly realize is that our modern life, and really over the last, I'd say 75 to 80 years, has really been changing these ecosystems. So these different parts of our body with different microbiomes have been altered because of our environments. So kind of to illustrate this, I wanted to kind of quickly go through a short timeline of some of the major changes in our environment and kind of suggest how those may have changed our microbiomes. So in the 1940s, a lot of times I guess we don't really think about some of these things, it seems like it's been around forever, is when we really started having refrigeration in most homes, at least in the US. So of course refrigeration then meant we could buy foods that were could be stored for longer periods of time, so not as much fresh food. So you got more processed, more packaging, so the food itself was a bit different because we could refrigerate it. Also around that same time, or in the 1940s, the urban to rural living was about 50-50. Now it's closer to about 82% urban, and that of course changes our interaction with the natural world around us. And then in the 1940s is when antibiotics were first introduced. Penicillin was the first antibiotic introduced and really got used a lot in World War II is when it first really started getting used. More, and then the early 60s where it became more widespread. So I think for most of us, we just feel like they've been around, you know, forever, but it really has been, you know, about 60 years or so that we've been using them. 1950s, there was a significant introduction of preservatives into food. Not to say there weren't some preservatives in foods before that, and there certainly have been, you know, natural preservatives used for a very long time, but a significant introduction of preservatives into food at this time. And so I'll talk a little more about that later. And also in the 50s and 60s, we started having a lot of antibacterial soaps and other antibacterial cleaning products that we didn't have before. And two particular chemicals, triclosan and triclocarbon, were first introduced into hospitals in the 80s and 90s. They started having widespread use. There's been a lot of studies done on those particular chemicals, and it wasn't until 2017 that the FDA ended up banning the use of those ingredients, along with 17 other ingredients that are oftentimes used in antibacterial soaps and washes, because they realize how toxic these chemicals are and how persistent they are, meaning that they stay in our environment, you know, almost permanently. We find these particular chemicals everywhere. They're in things like our shower curtains, and they were put in there initially, but they, you know, they don't go away. They seem to stick around and have a lot more risks than benefits at this point. Going back again to the 1950s, artificial sweeteners became widespread. Probably know there's been a lot of different types of artificial sweeteners, and what we know now is that our microbiome, especially our bacterial cells, they don't use these artificial sweeteners. These aren't beneficial to them. So we actually are influencing what bacteria can grow in and on us with a lot of these artificial sweeteners. Also in the early 50s, antibiotics in farm animals were started to be used not just for treating disease, but they realized that antibiotics in these animals were accelerating their growth. And so, of course, to people in agriculture, they felt like this was a big advantage because their animals were growing faster and made it to the time to be slaughtered and used for food much quicker. But we realize now that not only does it promote growth in animals, it's also promoting growth in humans. So in 1977, there were some regulations attempted. In 1997, the World Health Organization recommends antibiotic use in humans should not be used in animals. And again, 2017, when before we had a ban on many but not all antibiotics in animals for growth. So we are making progress in this area, but there's been a lot of antibiotics in the animals for a long time, and there's a lot of different effects from that, along with possibly connecting to obesity problems, but also just more antibiotic resistance in general. And the 1960s and early 70s is when microwave ovens kind of came into existence, and this, of course, also changed the way a lot of our food was produced and packaged and processed, oftentimes with a lot less nutrition. Also in the 1970s, high fructose corn syrup was developed and started being used in a lot of different products, and we know again that this influences the microbiome. So basically, changes in our food and our environments are being felt by these bacteria that live in and on us. And most of the testing that's been done by people like the FDA and EPA, whether it's a food product or a drug or other things in the environment, hadn't really taken into consideration the microbiomes of not only our own bodies, but the microbiomes of the environments, because there's microbiomes of plants and animals and soil that also need to be considered with some of the things that we're using. And it really had not been considered at all until fairly recently that we realized how much we're changing these environments and what effects that's having on us. So really our focus for the past 70 years or so has been to get rid of the bad microbes, and primarily a lot of this was on bacteria, but also viruses and some others. So with the processed foods here, when a lot of this, the processing wasn't all about getting rid of microbes, but there was certainly a way of packaging food so you could keep it around for a very long time by using food preservatives, for example, to allow you to keep it on the shelf for a long time. And that was to prevent the growth of bacteria and fungi primarily. Even having coatings on some of our vegetables to prevent antimicrobial growth. Obviously, things like disinfecting wipes and all of our different chemical products, that disinfecting products and cleaning products, that a lot of it was to get rid of potential pathogens, and a lot of it worked very well. Unfortunately, it was also getting rid of a lot of the good ones along with it. So this is a timeline that I picked because, for one, it kind of covered part of the time that I wanted to talk about, starting in 1960 and projected up to 2030. And this one is looking at specifically the prevalence of obesity among US adults. And so, of course, you can see this steady climb in obesity over this period of time. But I also wanted to point out that within this time frame, there's some other statistics that correlate, and one is that there's been a 176% increase in diagnosed diabetes in 1990 to 2014. This one is one of the most shocking, and this could even be a little higher now because this number is a little bit old, but a 400% increase in inflammatory bowel diseases since 1976. And a 10% increase in metabolic syndrome, 88 to 12. And then if you look down here, this is looking at from 1950 to 2000 and the emergence of a lot of different autoimmune and immune-mediated diseases. So things like Chrons disease, again, which would fall under this inflammatory bowel disease, type 1 diabetes, asthma, etc. So we've seen this increase in a dramatic increase in a lot of different diseases. Another thing I would put in here is just allergies in general. You see a lot more children with allergies that we didn't see before, and that actually kind of comes into the kind of the immune-mediated disease or diseases. And so these changes that I just went over in the timeline into our environment, and the increase in a lot of these diseases fit in the same timeline and also fit with the changes we're seeing in our microbiomes. So how and when do we acquire our microbiome? Basically, what's thought at this point in time, the consensus is still that we are born basically sterile, and we acquire our microbiome as we leave the birth canal, that as we leave the birth canal in our mouth, our eyes, on our skin, we acquire the bacteria that is in the birth canal. And so obviously there's going to be difference between normal vaginal delivery and a C-section baby, that they are not going to get the same bacteria at that point in time. And then from that kind of seeds our microbiomes to develop over the next few years, that along with other things that we're going to come in contact with in our environment. And there's a lot of studies on this, and I'll just mention a little bit on it, but there's been a lot of studies looking at the differences between babies that are born vaginally and ones that are born C-section, and we do see higher incidence of allergies in children that were born C-section, and some other diseases throughout their life, and you're looking to see, what kind of connections there might be with the microbiome there. And there have been some studies to even take C-section babies and swab the mother's vagina and kind of inoculate the babies to make sure that they are getting the correct microbiome. So about 3, we know that like the first 3 years is pretty much when you're developing your microbiome. So they were looking, you know, all the different places I talked about in your body. And also during that time is when you're developing your immune system, and your microbiome is really a huge part of your immune system, and is also training your immune response, because, you know, their bacteria, our immune system is supposed to fight, you know, things that are different than our own cells, but there's a training that goes on that basically our immune system learns to recognize what's the good bacteria and what's the bad bacteria, so that we're not, you know, in a constant battle with our own microbiome. And if children are not developing a microbiome, a good microbiome in those first 3 years, it's probably going to lead to other problems down the line, because things like autoimmune diseases and allergies. And we know that there's about 1/3 of the babies in the US that are born by C-section. And again, like I said, lots of different studies on this. One of the recent studies that I have referenced here shows that there are over 350 regions of our genome, so basically, you know, our genetic information in these babies that were born C-section that have genes that were turned off, and these genes are genes that we know are involved in immune response. So exactly how the microbiome ties into that, we're not sure. This is something that, you know, has been seen, that there is a difference in, you know, the genes being turned off that are involved in the immune response in babies born C-section in this study. And also, of course, just studies looking at the, you know, actual what composition of the microbiomes of babies born vaginally versus C-section, and we see there's a definite difference. Okay, so, you know, I've kind of already alluded to some of the ways that our everyday life affects our microbiome, but you really have to start thinking about, I think, that, you know, not only what you eat, what's in your environment, but, you know, what do you put on your skin, different lotions and things. We've gotten so used to using all these things all the time that, you know, we haven't maybe stopped to think about what effect it might have on these bacteria that we really want on our skin. I know one of the first things when the COVID-19 pandemic started and people started, you know, really wanting all the antiseptic types of gels and things of Purell, the first thing I said was, no, don't do that unless you have to, if you don't have soap and water, because you don't want to wipe out the microbiome on your skin. You know, that's supposed to be there. It's helping you to protect you. So using it when you don't have anything else, you know, once in a while is okay, but it's not something we want to be doing all the time. So we want to really think about, you know, what effect whatever we're using in our laundry soap, or, you know, in our what we eat, how it's affecting these other organisms. And this kind of goes back to a little bit to this hygiene hypothesis. It's actually been around for quite a while now. And it was a theory that suggests that we can really have too clean of environments. So this wasn't seen until, you know, in the last 50, 60 years. This generally wasn't a problem, and it's still not a problem in lots of the world where they actually, you know, have kind of the opposite problem. But what children need in the first 3 years is they have to have enough interaction with different types of microbes to stimulate or challenge their immune system so that it will respond to those threats and help the immune system to mature. So if they don't have that, if they're, you know, kept inside, if they're not interacting with any kind of pets, or they're not interacting, you know, outside with, you know, soil bacterium and things that you find out there, we're finding that those children end up with more allergies, asthma, and autoimmune diseases later in life. So it's really important that they are getting this interaction early in life, and that first 3 years is super important. So this kind of just is continuing on with that with some other studies. So, you know, exposure to bacteria and or their endotoxins, which are certain bacteria types, could confer protective effects towards allergy development. So if you think about it, if your immune system is supposed to be looking for things that are foreign and reacting to it to get rid of it, and if you're not being exposed to enough foreign, what we call antigens, that your body is going to develop an immune response to, your body oftentimes will start reacting to things it's not supposed to react to. So it will start, reacting to, the cat or the dog or different foods or things that, maybe you would have been fine with, but you'd almost think like the immune system's bored, right? If it doesn't have enough interaction from other things in the environment. So it starts reacting to things it shouldn't.
Super user is the person that gets all the 20 licenses.
I'll have a sip of Coke.
Kind of like the UH site, and
Again, I think I'm muted still. Okay. So again, pet exposure is one of the ways that they've found you can actually risk or lower the risk of some allergies. So they've seen that a lot of children that grow up in rural areas and on farms and that have a lot of interaction with animals on the farm actually seem to have fewer allergies and asthma. And of course, breastfeeding, human milk, we know is both what we call prebiotic and probiotic. So probiotic means that it contains some beneficial bacteria, and prebiotic would mean that it contains nutrients that would help to feed those beneficial bacteria. So what does having what we consider a healthy microbiome do for us? As I've already mentioned, it's a significant part of our immune system, both its development and also the action of the immune system. It actually, the bacteria helps to moderate the immune response and helps to direct a lot of the immune response. It also helps in things like digestion of our food and production of different vitamins. We know that the gut actually produces more serotonin than the brain, and that there's certain bacteria that are involved in kind of inducing the production of serotonin. So this has led to some studies on looking at the changes in the microbiome and diseases like depression. And this is a link here. So I know she's going to post this, but this is a link here that you can go to on more on that. They've also found links to a number of different things like sleep patterns, exercise amounts, response to certain drugs, and metabolism. So in other words, people are looking at what's your microbiome and then some other aspects of your even personalities and kind of things that you're more prone to, like how much sleep you need, to see if there's a tie-in with the microbiome. And they are finding that in a lot of cases there is. So a lot of things that we put more to our own genetics is actually probably the genetics of the microbiome. Good news on that is we can probably change the microbiome, and therefore its genetics a lot easier than our own. Okay. This is a short list of diseases that have been shown to have an association to the makeup of the microbiome. Some of these I've already mentioned. And when I first started talking about the microbiome, and I guess still when I talk to people that haven't heard much about it before, I always feel like I'm selling snake oil because, it's like, it's connected to this and this and this and this and this, all these things, and it doesn't seem possible. But the more you study it and understand, what a vital part of us it is, it really does make sense how it can have impacts in all these different areas. And we're finding very specific mechanisms as to how this happens. It's not just a association or a correlation. We see causation, which of course is important in any kind of studies like this. Okay, let's see if I skipped one. Okay. This I already mentioned, just another slide on the immune system, that it's about 70% is in the gut, and a lot of this has to do with our microbiome. We know that it has a big role to play in regulating the inflammatory response, and this is something, if you've been reading much on COVID-19, and they talk about the cytokine storm and things later in the infection, that's a lot of inflammation. That's a lot of the inflammatory response. And so we know that it helps to regulate that inflammatory response, and that can be a good thing, and it can also be a bad thing, and things like allergies and asthma and cytokine storms. You know, you can have too much inflammation for sure. And so we also know that disturbances to the microbiome, so if we're changing it, especially early in life, can lead to some chronic inflammation, which can lead to things like Chrons and irritable bowel disease, different cancers, diabetes, and Alzheimer's, just to name a few. So just touching on a couple of those that I mentioned. Depression's been a big one, a really important and interesting area of study with this. And you know, this is a little bit of an older one, but this is actually 2017, this study, and this idea of the gut feeling. And I always thought this was a great way to think about it, because I think we're all very familiar with the, you know, feeling of butterflies in the stomach, or if you're angry or you're excited or you're sad. For most people, you feel it in your gut. Right? You know, your stomach gets upset, it hurts, or you at least have weird feelings in your stomach. And we know now that there's a, you know, direct line between what's happening in your colon, especially, and the effects on your brain. We know that the bacteria that live here in your gut are producing different types of chemicals. And some of that is leading to things like the production of serotonin that we talked about. And then that is going to the brain, which is going to affect mood, as well as other types of you know, chemicals that are being produced by bacteria and influencing other parts of our bodies, as well as in our brain, and certainly in our immune system. So we know this can be associated with, you know, gut inflammation, this chronic abdominal pain syndromes, and then eating disorders. All have, the microbiome seems to have a role in. And I'll come back to this a little bit later too, but you may have heard before, there's been a number of studies done using fecal transplants, and the idea with this is really to replace a person's gut microbiome with somebody else's, or at least to influence it. And they've been doing this really to fight one particular type of infection called C. difficile or Clostridium difficile, which happens to people who have been on a lot of antibiotics, oftentimes end up with this infection. And they had a very difficult time getting rid of it. And these people sometimes even wheelchair bound, they're so sick from this infection. And so quite a few years ago already, they tried the first fecal transplant to treat somebody with C. diff. And since then, there's been thousands of these done throughout the world, and with really high rate of success. But one of the things they started to see was that some of these people with the transplant, they saw like a change in mood. Like some of them might have were, you know, happier people and became depressed, or were depressed and became happier. Of course, if you're, you know, feeling better, you're going to be happier. But what they're finding is that it actually seems to correlate with the microbiome that came in with the transplant. So if it was somebody who was prone to depression, you might be actually giving that person a mixture of bacteria that led to depression. They also know with fecal transplants, they've seen you know, where you can have change in weight. So you could have somebody who has not had a weight problem, and that their microbiome being transplanted to somebody with a weight problem could actually correct that. They've done this a whole lot in mice, and have shown you know, the difference. But they're starting to see it in humans, and looking into the possibility of these kinds of transplants to treat some other disorders besides just the C. diff. So this is, yeah, another one just on obesity. There's a lot of different studies showing a correlation with the makeup of the gut and the mouth microbiome, which makes sense, right? They're going to be connected, and the inability to maintain a healthy weight. And all this is showing is just how the obesity rate has increased over, this is what, 1999 to 2016. So it's about 25 to 30% increase in both youth and adults in that time period. So also need to just say a little bit about antibiotics, because of course antibiotics are used to fight bacterial infections, are very good at doing that, but at the same time they're not that selective. And we're going to, when we take antibiotics, I think most of us are familiar with the fact that we are going to be affecting a lot of our other bacteria. As a matter of fact, for women especially, a lot of times when taking antibiotics, they end up with a overgrowth of our normal yeast that we have in our bodies, and other people will have this too. So we know we're disturbing the normal microbiome. We know that it usually takes, from the studies so far, about 2 to 3 months to recover your microbiome after taking a round of antibiotics. And we also know that antibiotic use in the first 6 months of life, when we're just developing that microbiome, has the greatest impact on the microbiome. And what's really bad is that more babies are given antibiotics than any other age group in the US. I think that's starting to change as people are understanding the impact. But I think it explains a lot of the increase in allergies and asthma and things like Chrons disease and irritable bowel disease over the last 40 years or so. And maybe even a lot of the depression. It seems like we're seeing a lot more mood disorders. Okay. Just some fairly recent studies on cancer treatment in the microbiome, and there's other ones out here on other types of disease treatment, and I'm sure there's going to be a lot more. But these were 2 studies about a year ago that looked at the types, basically looked at your makeup of the microbiome and how effective a particular chemotherapy was for certain types of cancers. And they found that, depending on the microbiome, it seemed to have a big influence on how well you reacted to those drugs. So, as this says down here, the new studies add to a growing body of research suggesting that the composition of gut bacteria can dramatically affect how well cancer treatments work, and not just cancer treatments. I'm sure we already are seeing this with some other treatments as well. But this is, again, something nobody had thought to look at before, but could really explain at least some of the differences, especially when we don't see a difference genetically between people and how you'd expect them to react to a drug. Sometimes it doesn't make sense just looking at the human genes, looking at the bacteria, and we know how it interacts with a lot, things that we're eating and drugs included. It makes a lot of sense. Okay, and I already pretty much talked about this. So this is just using microbiome to treat disease. So the fecal transplants that I was talking about before. There's also now, they're starting to do microbiome banks in some places. So if if you're going to have maybe a surgery or something where you're going to have a lot of antibiotics given to you, or some other kind of therapy where like chemotherapy, that you might have a lot of killing off a lot of your good bacteria, that you can actually bank your microbiome so that it can be given back to you if yours is kind of destroyed or altered to a point where it's not working very well anymore. Also, of course, probiotics. We're all pretty familiar with that. There's and we're finding that some of these can be very useful. There you go. See, Trent has his right handy. And prebiotics, again, like I said, which are basically the food to feed the good bacteria. And I just thought this was kind of funny, but true, that this quote was, a colleague recently remarked that we used to live in a world where people got sick from exposure to feces and lives were saved with antibiotics. Now we live in a world where people are dying from antibiotics and their lives are being saved by feces. Obviously, that's a bit of a stretch, but at the same time, there's obviously some truth to that here, which is kind of ironic. And I'm certainly not saying, antibiotics certainly have their place, but we do need to be more conservative with them and think about their use more carefully. Okay, so this is something I just added in the last few days because I figured everybody, is thinking about COVID-19, and I thought it kind of interesting that I had not seen very much coming out yet about the microbiome because I'm sure there's a lot of people already looking at it. And so I started looking into it a little bit and just have been thinking about it. And obviously, just what like I had said before, being careful to not, disturb your normal microbiome by too much hand sanitizer, or definitely thinking about how you're eating and keeping your microbiome healthy because it is such a big part of our immune system. So we already know these things. It's been, it's well established. It's a significant part of our immune system. It's needed to train our immune system, and it's involved in signaling and regulating the immune response, both directly and indirectly. So all that is really pretty well established. So I just picked a couple of quick articles, and there were some, I actually went back quite a ways, a lot further than I expected on this, that I don't have up here. But this was one that's 2019, so it's March of last year, on virus and microbiota relationships in humans and other mammals. And so this is kind of a article looking at how our microbiome and viral infections interact, and how basically this was one of the figures from this article that I thought was kind of interesting, showing the normal microbiome here. So this would be found in your gut, and how they promote different parts of the immune response, which are showing here as inflammasomes and dendritic cells, which may not mean a lot to a lot of people, but it's part of our immune response and a lot of our innate immune response. And how that then can go, and this is with the influenza virus, how it can go and actually defend our bodies from the influenza virus. But then at the same time, the virus causing reactions in other parts of our immune system, and that leading to changes in our gut microbiome. And basically, what it looks like is that, on one hand, we have our good microbiome trying to fight the virus through our immune system, and the virus trying to shut down the microbiome that's trying to fight, that's trying to induce a good response. So it's just this interaction, that hasn't really been looked at too much before, that how the virus and our normal microbiome are kind of in competition with each other. So that, also leads to, our understanding of that's going to help us on thinking about how do we, fight different viral infections. If we understand that it's shutting down our microbiome and changing it, maybe not shutting it down, but changing it, and doing that is changing our response. Maybe if we can, either stop it from shutting this down, or, add to the microbiome with things like probiotics, maybe we can prevent this from happening. And this was also in July of 2019. Commensal microbiota just means these are the bacteria that live in and on us that are part of our normal microbiome, that are generally, beneficial. They're certainly not a negative, and viral infection. And this is a review of all the different research that's been out on this. And this is a quote from this article. It says, a plethora of evidence suggests that the commensal microbiota regulates and is in turn regulated by invading viruses through diverse mechanisms, thereby having stimulatory or suppressive roles in viral infections. So just what we just looked at, that our normal microbiome is helping to shut down the virus, and at the same time we see some viruses also sometimes even using our own immune system to shut down the microbiome. Furthermore, the integrity of the commensal microbiota can be disturbed by invading viruses, causing dysbiosis, which just means a change in the microbiome in the host, and further influencing virus infectivity. So these were just two articles I picked. There's actually a lot more, but this, I think the take home on this, I have to keep moving this bar around. is that a healthy microbiome is essential to the strong immune response, but pathogens like viruses can influence the makeup of our microbiome and prevent its function in the immune response. So maintaining a healthy microbiome is a key factor in immunity. So a lot more work's got to be done to understand those specific mechanisms that are involved, and this could, definitely lead to new treatments for a lot of viral diseases. I don't know if has there been any questions written up? Because I have not looked.
Yes, there was one question about the numbers in one of the earlier slides. Could these numbers partly be from better reporting and testing of those things, or is it based upon percentages of people tested, etc.?
So I'm assuming they're talking about probably this real early slide.
Yeah, it was related to the higher numbers of allergies and autoimmune disorders.
And so could it be just from, I think you're always going to have that as a factor, right? I mean, just, the numbers, but what the studies that I took those from, they were showing a significant, increase, so the numbers were significant. So the population sizes were large enough that they were looking at that they could state this as a significant increase. So yeah, with more numbers, you might find more, you might find less. But in those studies, it was a significant increase. I'm not sure if that totally answers that.
I was just participating.
Oh.
There you go. There's a question.
Thank you.
I have a verbal question.
Sure.
So I was listening to a podcast probably years ago now, and they were this wellness MD was talking about how a lot of his patients that suffer from, depression, a lot of autoimmune disorders, they were finding bacteria that was supposed to be confined simply to the large intestine in their distal portions of their ileum, their small intestine. Are you able to speak to that at all?
No, I've heard of that also, but I haven't seen anything really conclusive on it.
Okay.
But yeah, I mean, and then the things like leaky gut syndrome also.
Right. Yes.
Yeah.
Okay. Thank you.
Yeah. Okay. Let's go on from here. So I will just briefly talk a little bit about some of the work that we've done here. Just I have a little summary of this because it's too long, but this started with sabbatical work that I did on food preservatives and the effects on the microbiome back in 2014 when I was on sabbatical at the Buck Institute. And basically there I observed a significant decrease in the diversity of gut bacteria in fruit flies is what we were studying that were fed with sulfites. So I had picked sulfites for a variety of reasons, actually some preliminary data that I had got. But it's a food preservative that's in a lot of different foods. So just a little short history on sulfites. One of the things when I started studying this that I just found so fascinating was that it was back in 1958 that the FDA generally regarded as safe or gave that rating to sulfites and actually a number of other different food preservatives. And a lot of the rules and regulations regarding food preservatives are still based on these studies that were done in the late 50s and early 60s. So that was a bit disturbing. And then I won't go through all of these, but you can see there's been a lot of people evaluating and reevaluating the use of sulfites and how much are actually safe. And they have changed how much are allowed in foods and which foods they're allowed in as new information has come out. So today, the World Health Organization, this is what they say as the allowable daily intake, or the ADI. And of course, one problem with the allowable daily intake of anything is, this is based on how much for a 150-pound adult. Obviously, we're not all 150-pound adults. And so even if this is very accurate, this is assuming that you're eating a certain amount of food with that, whatever the chemical in it, and that you're not eating 3 portions of that, or you're not eating that along with 2 other things that also have that same allowable daily intake, or that you're a 50-pound child, or there's all this variability. So to really be able to have a safe, allowable daily intake is very difficult to decide what that might be. There's certain foods that the sulfites have been restricted from because of it will destroy thiamine. It does occur in low levels in some fermented foods. They now, if it says there's sulfites in your food, it means that they have concentrations over 10 parts per million. But we know that it can go up much, much higher than that, and you don't really know what it is. You just know it's over 10 parts per million. Okay. So these are some examples of some common foods that contain added sulfites, so not just naturally occurring. And of course, the one that I usually get asked about the most is wine. People are like, oh no, and it is actually, white wine contains quite a bit more generally than red wine. So if you're a red wine drinker, you'll be happy to hear that. And you can find some wines that will say no added sulfites or no detectable sulfites, but most of them do have sulfites in it, and it really ranges in the amounts of sulfites. And then a variety of other types of products. Real lemon juice, this is one of them that really surprised me, has 3 kinds of sulfites in it. And there's coconut milk, canned coconut milk that has sulfites, and there are other ones, brands that don't. So you have to really look for it. And with a lot of these foods, you can find some that have it and some that don't.
I've seen a product that advertised they could remove sulfites. It's something you poured your wine through. Is that at all realistic?
When I first started doing the studies, that was one of the first things I said to Peter Fisher, my colleague that works on this, he's a chemist. I said, we should come up with a way of removing the sulfites from the wine. This would be a great product. And not too long after that, you started seeing some. And I think that the thing is that it could possibly work. I think a filter would work the best. But I know there's been one out that is actually hydrogen peroxide. And so obviously you don't want to be putting a lot of hydrogen peroxide into your wine. Not only it probably wouldn't taste that good if you had very much, but also you don't really want to be drinking it. So, I think there's some potential there, but I haven't seen anything that seemed like a really good solution yet. Probably need to still come up with that. These are just some of the common observed reactions to sulfite. A lot of it's digestive types of allergic responses. But it can be pretty severe, and actually have had deaths from sulfite exposure. But those are people who have really extreme responses to it. But that has changed a lot of the regulations on it because of those kind of responses. So our first set of studies that we did at the college ended up in this paper back in 2017, looking at sulfites and whether or not they inhibited the growth of 4 beneficial gut bacteria in concentrations that were considered safe for food. And we found, of course, that it was inhibiting their growth or killing them off. And this slide is just looking at some of the health benefits of the bacteria that we tested. So obviously we wanted to pick bacteria that we knew a lot about, that we knew were beneficial, and to see whether or not sulfites would have an effect on this. But all of this was done, outside of the body in a dish. So it was in vitro. Of course, there's always, there's a lot of questions that are, would the same results happen in the body, which we could not answer. But kind of the summary of that was that sulfite preservative ingestion may be connected to significant changes to the gut microbiome and subsequently several diseases or conditions associated with altered gut flora. Really much in the same way as antibiotics are. We've already seen these connections with antibiotics and these types of connection to different diseases because of changes in the microbiome. And if you think about how many preservatives are in our foods, in the last 60, 70 years, and how, you know, for a lot of people, this is a daily thing that you're introducing these into your gut, that the idea of that altering your gut microbiome is pretty easy to see how that could happen. So after that set of studies, we really wanted to do something in vivo in the body. We wanted to see how these sulfites were interacting with our microbiome in the body. And the easiest place that we could think of to do this, other than the skin, but because we wanted it more connected with the digestive tract, was the mouth and saliva specifically. So we started a couple years ago doing studies on the effects of not only sulfites. We've looked at a little bit of some other preservatives, but mostly we focused on sulfites again, on the microbiome in the mouth, which is actually a pretty significant microbiome also, but hasn't been studied as much as the gut microbiome. But some of the studies that have already been out show the connections to the health of the mouth, you know, and the health of the microbiome, or what microbiome you have in your mouth, and diseases in other parts of the body. So they're actually even starting to look at your mouth microbiome to predict possibly some diseases that we find in the gut or other parts of the body, that we can see changes there sometimes first. So diversity and source of healthy bacteria of the mouth is going to be influenced by, of course, your diet. It's going to be a big one. Your genetics is going to be some of it. We found gender to be, or other people have, and actually we are seeing that too a little bit, might moderate it, and also age, and also things, of course, like smoking. And then another reason we picked it, of course, is just it was easily accessible. So some of the questions we wanted to ask are how are individual saliva samples alike or different, not only between individuals, but also taking the same individual saliva over, you know, like 2 or 3 month period, and see, did the bacteria types or numbers change in their mouths over time. So we wanted to look at the bacterial cell number and cell types, reaction of those bacterial cells to sulfites, and also wanted to look at some of the enzymes that are in the saliva naturally, like amylase, which breaks down starch, and lysozyme, which actually breaks down the cell walls of different bacteria. So this certainly has a big influence on the microbiome. We wanted to know whether or not different people had different levels of lysozyme activity or amylase activity in their mouth. And again, we've mostly focused on lysozyme here, as well as wondering, did sulfites maybe interfere with some of these enzymes? So actually, let me go to this one first. This is actually from a year ago, and we've done a lot more on this since then, but it's kind of just supported this. And I know for a lot of people, this may not really look like much of anything, but you can probably see there's 5 individuals here in the different colors, and this is their activity of lysozyme, is what we're showing here. And so this is kind of like baseline. And the biggest thing what you can see is that different individuals have different baseline activity rates of this enzyme. And this is important in part because as far as we know, nobody's actually looked at this. We don't see any studies where they've tried to look at baseline activities of lysozyme, and we know it's going to influence the microbiome. And then this is actually showing that sulfites decrease lysozyme activity in human saliva. So up here is without sulfites, and here down here is with the sulfites. So you can see that we reduced the activity of the lysozyme with sulfites. So that could also have an influence on the microbiome. And this was one of the ways that we looked at the composition of the microbiome was by actually isolating the bacteria that were there, and before and after treatment to sulfites. And then we narrowed it down in this. We were doing DNA sequencing studies, and we narrowed it down to these groups of, in this particular experiment, to these groups of bacteria that were the most prevalent in those samples, because there was lots and lots more. There's hundreds of different bacterial types. And so all I really want to point out here is if you even just look at these first 2, these are each a different individual's saliva sample. Whoops. And this is untreated, and this is treated with sulfites. So each of those are treated and untreated, and basically an immediate reaction. So the differences that you see here, we think are due just to the sulfites, so that we are seeing changes, and these are percent of the total of that particular group of bacteria. So some of them you don't see real drastic changes, some of them you see quite drastic, but we're talking, this is immediately. This is like in the first 5 minutes of exposure. So it doesn't take a long time, it seems, to see these changes. And since then, we've actually done quite a bit more sequencing studies, and we've gotten the way that we're going about testing the saliva samples, you know, how we're treating it and everything is, we've refined it to where I think we've got it down to a much more accurate. And so those are kind of old, what we just looked at. We're, you know, I think we're going to see some more significant types of effects than we were initially, although we certainly were seeing some. So our conclusion so far has been that sulfites affect lysozyme activity, and it seems to inhibit to a certain degree the sulfite lysozyme activity in the saliva. They definitely affect the number of bacteria and the types of bacteria. So we are changing the microbiome with the sulfites pretty immediately. And that helps us in our understanding, and when I say us, I mean the scientific community, of environmental factors that influence the microbiota, and quantitative analysis of sulfite effects. So really looking at numbers. So foods containing sulfites, again, we're concluding that play a role in altering the mouth microbiome and may ultimately affect the overall health. Okay, so kind of the last bit of this, I think, is, you know, just some things that help you think about what you can do to help improve your own microbiome or to keep it healthy. So a lot of things we've really already gone through, but not taking antibiotics unless they're, you know, make sure that they're necessary, and then take them correctly. So don't overuse them, or, you know, take only partial bottles and then get rid of it, that type of thing. Possibly the use of probiotics and prebiotics, and there's more and more evidence on that, and I'll have another slide that talks a little more about that, how to pick those. Avoid preservatives and processed food. Look at the label, you know, see what's in it. If there's a lot of stuff on the label that you have no idea what it is, it doesn't even sound like, you know, something that's a food item, you know, maybe avoid that, or try to pick a different brand. You know, really try to get fresh foods, eat a lot of fresh foods, and fruits and vegetables, a lot of plants, right? Plant-based is definitely what people are finding is the best. Avoid meat from animals treated with antibiotics or hormones. This is pretty easy to find now, at least in the US. Most of them will say right on it if they haven't been treated, because we know those are affecting our microbiome as well as some of our own cells. And eat more fermented foods, so yogurts and kombucha and kimchi, and anything fermented like that has some natural occurring, you know, bacteria and yeast and things in it that can be really beneficial. And foods with fiber, so basically a plant-based diet. And just avoiding unnecessary chemicals in your life, really starting to think about everything that you are using. There's so many things we've gotten used to using every day that are really unnecessary and could be doing damage not only to our microbiome, but also to the environment, and just, you know, things that were like dryer sheets that you use and then you throw them away. Right, you know, maybe use them a couple times and throw them away, and people kind of got by without those for an awful long time. Probably don't really need them, and there have been studies to show that using dryer sheets basically kind of coats your clothing or your towels, and then when you have that on your skin, you're actually disrupting your skin microbiome.
There's alternatives now too. I know that's something me and you have been looking into.
Right.
Dryer balls made out of wool, which still help reduce drying time, and don't have all this.
Right, yes, that's right. Yeah, so the balls that can actually help to kind of soften and take out static cling and stuff, but without any chemicals. Yeah, thanks. Good suggestion. And And then just as a society, we have to push for developing new narrow spectrum antibiotics instead of all the broad spectrum that just kind of kill off everything. Stopping the overuse of antibiotics in farm animals, which is happening. Discourage unnecessary C-sections and encourage breastfeeding. And then just choosing a probiotic. Some of the things that I've found, this is one of the things I started off really early on in these studies, is I really looked at a lot of probiotics and tried to identify some that seemed to be better tested and what seemed to be important in a probiotic. And one is having at least 8 to 10, I'd say, different species or genus. And they should list them, on the bottle. It should say it right on there. I've seen some that don't, and that definitely makes me suspicious. Look at the company that sells them, see if they have any peer-reviewed science or clinical trials behind them. Some do, and most don't. If you have a specific issue you're trying to treat, look up which bacteria are known to be helpful, and then look for probiotics with that bacteria. So in other words, just say you're trying to treat depression, you can look up depression and microbiome, or depression and probiotics, and you can actually identify bacteria that they've found to be important to help alleviate, in some cases, some depressions or anxiety symptoms. Make sure the bottle lists exactly what kind of bacteria, as I said, and then mounts on the label. Some other things in this comes from research that's been done. It seems to be a little bit better to take at night before bed, because we know that the numbers go down in our guts at nighttime. We don't have as many of the bacteria there, and so if you're trying to introduce something new, it's easier to get it established at nighttime than during the day when you might be outcompeted by all the other ones there. Unless it really bothers your sleep or something, then I would say take it in the morning. I always, for any kind of thing like this, I'd say just take one a day to start, and then even if the package says to take more, wait a week or two before taking more, and then only if needed, because again, just like the allowable daily dose or intake of different preservatives, everybody's going to be a little different, so to say everybody should take 2 a day doesn't make sense to me. That different people are going to have different reactions, so take less to start. If you're taking antibiotics, then take the probiotic in between doses, or wait until you're done with antibiotics to start them, because of course you're going to be killing off a good chance a lot of the probiotics. But you also want to refurbish the ones that, the good bacteria that the antibiotics are killing. And then another thing that I've found in the last 8 years or so has been this beneficial probiotic yeast that's called Saccharomyces boulardii. And this is a really good thing they've found to take along with antibiotics. It's not affected by the antibiotics because it's not a bacteria, it's a yeast. And it basically will help to maintain the lining of the gut and encourage, the good bacteria, kind of help them to maintain and while you're taking antibiotics, or to repopulate afterwards. This Florastor is one that contains this type of yeast. And one of the reasons I recommend this one is because it actually has been involved in a number of clinical trials and shown a lot of success with this particular probiotic. It's really useful for like traveler's diarrhea, and it also seems to help prevent the C. diff that we talked about that happens, that people have gotten the fecal transplants for, so the bacterial infection that comes with oftentimes taking a lot of antibiotics. These are a couple other ones, and I'm not, I don't have stock or anything in any of these. I probably should, but these are just some that I've researched to feel like they are pretty beneficial. And sometimes they'll have, numbers associated with it that shows that they're, they're really a little more linked to specific strains of a bacteria that have been tested. So this is one of the good ones. This is also one that for looking at anxiety and depression, that there's a particular couple of, this one only has a couple bacteria, but these are have been studied to show that they were beneficial, at least to some people in these depression studies. And then this is just kind of saying, look at, it sounds so good, just this coconut oil, cocoa butter, and African shea butter. This is just a cream, a hydrating cream. But when you start looking over here, you realize maybe this isn't what you want to put on your skin. I mean, it sounds awfully good over here, but just to look at, the ingredients. And the same, with these other products like I was showing. This is the ingredients, and this one has the metabisulfite. Okay, and just the last couple of slides, I'm not going to go into too much, but these are just a couple of the big microbiome projects that have been going on for a while. This is one that was called the Human Food Project, and they've been looking at basically microbiomes across the world, and then what people are eating and what kind of diseases they do or don't have, and just trying to connect the dots type of thing. And let's see, and then the Human Microbiome Project was one of the spin-offs of the Human Genome Project, and it started all the way back in 2007, and the goal was to characterize the human microbiome and determine its role in human health and disease. So it's led by the National Institutes of Health, but it's an international consortium, so it's going on around the world, and so you can go and see some of the things they're doing there. And this is actually their website here. So that is pretty much all I had. Does anybody have any other questions right now? Comments?
So do you recommend I find the happiest, thinnest person I know and ask them for a food sample?
Oh, it's a thought. Yeah, no, I think that's going to be happening. Not too far in the future, really, because they're certainly seeing this already, and they're starting to look at that. And actually, I saw it with the COVID, some of the COVID studies that with the fecal transplants that they're now screening for that, because we know that the virus is found in the feces also. So they're starting to screen for that before they do the fecal transplants. So they're not introducing COVID-19 to somebody who's coming in to get rid of Clostridium or something. Any other questions? Or I don't think there's any in the chat. So, okay. So just like in class, nobody wants to come on and ask the questions.
Answered all my questions.
You answered all. Okay. Yeah, I think you already had them, Emily. But thanks again for everybody for coming. And if you have questions later, you can always email me too. Yeah.
Thank you.
Thanks.
Thank you, Sally.
Thanks, Linda.
Thank you, Sally. And everyone, please look for the survey evaluation. Thank you.